So in the last video, we looked real briefly at some of Gregor Mendel's experiments with plants and how he was able to observe this pattern of inheritance that we now understand better in terms of modern genetics to be the way that meiosis really drives the reshuffling of alleles. So what I want to do in this short video is to talk about the first of two really important laws that Mendel kind of teased out, but we're going to talk about them from kind of a modern view and a modern understanding of the chromosome and genes and alleles. So the first is called Mendel's law of segregation. And the first principle or point to this law is that alleles, which are different nucleotide sequences coding for the same character, account for the different traits that we observe. The difference between, for example, a purple flower allele on one chromosome and a white flower allele on its homologue might be as little as one base pair difference. Often it's more than that, but it doesn't take a whole lot of change in the genetic code to ultimately lead to an altered protein and ultimately lead to an altered phenotype. So in the last video, we were saying that purple flowers are often dominant to white flowers, and so this would be an example here of a heterozygous condition where genotypically we'd have big P, little p as the genotype, one dominant allele, one recessive allele for a given character. But phenotypically, we would have the dominant trait being expressed. And that's how the concept of dominance and recessiveness plays itself out. And many genes do follow a simple dominant recessive pattern, though not all of them do. So the second point in Mendel's law of segregation is that diploid organisms, you and me, plants, most complex eukaryotes, have two alleles for every character. Why is that they have two? It's because they're diploid, meaning they have chromosome number whatever, let's call this chromosome 4, that they got from one of their parents, and chromosome number 4 that they got from another parent. All the other chromosomes are present, we're just tracking this particular chromosome, and all the other genes on these chromosomes are present. We're just tracking this particular gene that codes for the character of flower color. So diploid organisms have two alleles. Sometimes they're different, like this heterozygote. Sometimes they're the same, like in the homozygotes. And the same could be homozygous dominant, could be homozygous recessive. But there are always going to be two alleles, because that's what it means to be diploid. Now, let's tie this in with meiosis. This is really important. In meiosis, the main thing that's happening is that homologous chromosomes, so chromosomes that map out the same, code for the same characters from two different sources, a chromosome 4 from mom, a chromosome 4 from dad, end up being separated into the gametes. That's the whole idea of reduction in ploidy. A diploid organism has two of every chromosome. A haploid organism has one of every chromosome. Because the homologs are carrying these separate alleles, and because the homologs get separated into different gametes during meiosis, the alleles also get separated into different gametes during meiosis. So for example, in this picture here, let's say this is chromosome 1 for some organism, and at a particular locus, particular location, an address, if you will, on chromosome 1, and the reason it looks like an X is because it's undergone a DNA replication, so each one has been doubled identically. At a particular address on here, you're going to find the gene that codes for, let's say, flower color, and maybe this is the recessive flower color allele, and one nucleotide difference here, you've got the dominant flower color allele. And this whole chromosome came from one parent, and this whole chromosome came from the other parent, and they are getting separated into the two new daughter cells, which will ultimately lead to gametes, and therefore the two alleles, for example, the dominant, or that we said, I guess this was the recessive white allele, and the dominant purple allele are being separated into those gametes as well. Make sure that connection makes sense to you through this law of segregation before you get too far into trying to understand Mendelian genetics and trying to carry out these different genetics problems. So if we think about this, so here's a progenitor. This could be a stem cell, which is going to be diploid of that particular plant. And it's got on one copy of chromosome 1, it's got the big P allele, and the other copy of chromosome 1, it's got the little p allele, right? One came from its mother, we'll say, and the other came from its father. It's a heterozygote genetically, or its genotype is heterozygous, and we've drawn it big P, little p to imply that this is a simple dominant recessive relationship, and therefore the phenotype is going to be purple. Right? The dominant allele is going to get expressed, the recessive allele is not going to be expressed. Now, when this cell becomes a source for forming gametes, half of the gametes of this organism will carry the dominant allele, and half will carry the recessive allele. You notice what happens to the genotype. The genotype of the somatic cells of the organism are heterozygous, and they're diploid. The genotypes of the gametes are haploid. They only have one or the other. And so we don't use terms like homozygous and heterozygous, but we can say dominant and recessive. That's the idea of the law of segregation. Now, even though we're showing it as if there's one big P in this, let's say this is a sperm cell, understand that every other gene is represented there. The entire genome is in there. We're just tracking one gene at a time. We can't track all 25,000 at a time. And then the fourth principle from Mendel's law of segregation is that in diploid organisms, many alleles are either dominant or recessive. This isn't 100% of the time, but it's common enough that it's worth learning, and you'll learn other patterns of inheritance as you move forward in genetics. And so the difference between these two alleles coding for the character of flower color, like I said before, may be as little as one base pair difference, but that one base pair difference may be all it takes to make one dominant and lead to one phenotype and the other recessive, and potentially lead to the other phenotype in the homozygous recessive condition. So the homozygous recessive condition, little p in this case, is the only time you're going to see the recessive phenotype in a dominant recessive relationship, meaning that both gametes that fused were fertilized together to form the zygote had to both be carrying the recessive allele. If either one is carrying the dominant allele, you're going to end up with the dominant phenotype, and if one carries a dominant allele and the other carries the recessive allele, you'll get the dominant phenotype and a heterozygous genotype. If both are carrying the dominant allele, you'll still get the dominant phenotype, but it'll be a homozygous dominant genotype. So make sure you think through all those possibilities in this kind of simple scenario where we're tracking only one character at a time, one gene at a time, with two simple dominant recessive alleles, before you try to get into more complicated systems. Now, to visualize this a little bit, let's say that this blue chromosome represents chromosome number 10 that you got from your mom. Maybe you didn't get it because we're talking about white and purple, but a plant got from one parent, and let's say this red one got from the other parent, right? These are chromosome 10 and chromosome 10. Okay, and they're practically identical to each other. They're homologs, they're not identical, they're not perfect copies of each other. And at a particular location, if we were to map these out, we would find a gene coding for flower color. And if there's any nucleotide differences in the sequences, we're going to have different alleles. And so in this case, we've got a purple allele, and we're labeling it big P because it's dominant over the white allele, which is little p, so our genotype is heterozygous. This would be prior to DNA replication, to S phase of the cell cycle, because in S phase of the cell cycle, we'd have two copies, identical copies of each of these. So we'd have two big P's connected at the centromeres, two little p's connected at the centromeres. And in the first round of division, so here's our diploid stem cell, here's chromosome whatever, 10 I guess, right? And one of them from one source is carrying the recessive allele, the other from the other source is carrying the dominant allele. All the other genes are there. We're just tracking this one chromosome right now. DNA replication takes place, and the identical copies are linked together at the centromere. In the first round of meiosis, meiosis 1 ends with those homologs being separated. And so that means the alleles that we're tracking are being separated as well. So in this case, half of the the gametes would be carrying the recessive allele and half would be carrying the dominant allele. The second round is much more like mitosis, where the sister chromatids are separated, but it doesn't matter. You still end up with half recessive allele, carrying the recessive allele for the trait you're interested in, and half carrying the dominant allele. So we start with a single diploid stem cell that has the diploid genotype of the rest of the organism, the somatic cells of the organism. And in the end, we end up with 4 haploid gametes. And the gametes have to be haploid, don't they? Because gametes come back together and fertilize in sexually reproducing organisms, and that restores that diploid state again. If ploidy was not reduced from diploid to haploid and we had diploid gametes, then we would end up with quadruploid or whatever it is, 4 ploidy in the offspring. And that's obviously not going to work. And then on down the line with every round, we'd end up increasing the ploidy, and that wouldn't be a sustainable situation. So let's finish with you clicking pause on this video and thinking through this concept check. Make sure you understand that before you move on to the next video, and if you need to watch this one again, please do. So now's the time to hit pause.